Hi everybody. In our last video for organic compounds, we're going to be looking at proteins and nucleic acids. Now in the first video that talks about organic compounds, we went over what it meant to be an organic compound, what we meant by complexity or large molecules, simple molecules, biomolecules, macromolecules, dehydration synthesis, which is how our body makes things, and hydrolysis, which is how our body breaks things down. And then that video also delves into carbohydrates. In the second video we did, we went over lipids and why lipids are so important. And if you remember, we have 4 different macromolecules or biomolecules. We have carbs, which are sugars and starches. We have lipids, which are fats, oils, and waxes. And then we have proteins and nucleic acids. And they're listed in this order according to their complexity. And when we talk about complexity, we're talking about bonds. How many pieces do they have stuck together? So carbohydrates are first on the list. That's not a lot of pieces. That would be like having a little Lego person. And then lipids are second on the list, so they have a few more pieces to them. It might be like a little Lego car. What we're going to look at today are proteins and nucleic acids, which are incredibly complex. Lots of pieces to them. So we're looking at like a castle in the Millennium Falcon. So in the case of proteins, when we talk about your genetics, we're talking about proteins. This is why proteins are so important to cells. There are a whole bunch of different types of cells. I mean, if you think about humans, we have skin cells, blood cells, stomach cells. Well, what makes each cell different is the protein those cells make. So proteins are the recipe, are the products of recipes. So when we talk about DNA, which is a nucleic acid, DNA is a cookbook. And it has recipes in it, just like your cookbook at home has recipes that tells you how to make different things. Well, your DNA is this huge cookbook, and it has recipes on it, and that's what our genes are. So all of your life you've heard the term gene, but you might not have known what it was. So when we say I have the gene for brown eyes, that means I have a certain recipe. And what genes tell us to do is to make proteins. So if you have the gene for brown eyes, you have a recipe that tells you to make a certain kind of protein. And those proteins give you brown eyes. So proteins are incredibly important, and they are, third on the list. Well, in the case of carbohydrates are made from carbon, hydrogen, and oxygen. In the case of lipids, they are made from carbon, hydrogen, and oxygen. Proteins are going to have an extra ingredient to them. And you can see those ingredients here in this picture of what's called an amino acid. You'll see carbon, hydrogen, oxygen, but we also have nitrogen, which is the letter N in proteins. Now we've been talking about monomers. Monomers are the Legos that we use to put things together. In the case of carbs, our monomers are monosaccharides. That's the smallest type of sugar you can have. In the case of lipids, our monomers were glycerol and fatty acids. In the case of proteins, proteins are made from amino acids. Amino acids are the Legos that we use to put together to make a protein. Now, in the case of proteins are always drawn to look like a pearl necklace or a beaded necklace. So anytime you see a circle with an AA in it, that stands for amino acids. But amino acids are the beads that we string together in order to make a protein. And all amino acids will have 3 parts to them. Okay? And they get their name, amino acid, because they have, if you'll notice right here, they have an amino group. And over here, they have a carboxyl group. Well, carboxyl, by having a carboxyl group stuck onto something, it makes it an acid. So this is where the term amino acid comes from. So every amino acid in nature has the same 2 structures. Also, every amino acid in nature has a carbon-hydrogen backbone. Well, what we find, again, our amino acids are made from carbon, hydrogen, oxygen, and nitrogen. So now it has a 4th ingredient. So that's even more Legos. So we can build something even more complex and something even bigger. Well, there are 20 different amino acids in nature. And it's like having 20 different colors of Legos or 20 different colors of beads. And nature will line those beads up. And just like you have a pearl necklace, there's a string in between each bead that holds the beads together. Well, when we make a protein, we have to hold these beads together, and the beads are the amino acids. And so the beads, the string right here that's between them is what's called a peptide bond. So you'll sometimes see peptide bonds or proteins also being called polypeptides. And you are what you are based on your proteins. So as humans, our DNA has some of the same recipes. So we all make a stomach, we all make bones. But some of our recipes are a little bit different, and that's what makes each of us different. But like I mentioned, in nature, there's only 20 different types of amino acids. Every amino acid has these three parts. Every amino acid will have an amine group, every amino acid will have a carboxyl group, and every amino acid will have a hydrogen carbon backbone. What makes each of the 20 amino acids different is this group right here, which is called the R group. Yeah, I know my picture might be over it. But the R group is what makes, it's also called the variable. It's what makes every amino acid in nature different. And there's only 20 amino acids, but there are thousands of proteins in nature. And so nature has had to get very creative with how it is going to make all of its different proteins. And so like I mentioned before, a gene is a recipe for a protein. So when you say that you have genes for brown hair, that means you have certain recipes and that allow you to make a certain protein, and that protein gives you more melanin, which is what makes our hair color, for example. But like I was mentioning, this is what a typical protein drawing would look like. Its proteins are always drawn looking like beads, and each bead is a different amino acid, and the string that holds them together are your peptide bonds. So again, a lot of times this is why proteins are also called polypeptides, meaning many peptide bonds. And all proteins, just depending on how many of those 20 amino acids you have and then in what order, you can make all these different types of proteins. And if you think about it, when it comes to baking, we don't have a lot of ingredients. We have vanilla, we have flour, we have baking soda, sugar, milk, water. But look at all the different things we can make. Cakes, pies, cookies. Well, it's all based on how you combine them together. So if we have an amino acid that is, let's just say ABC, A, B, C, with each of these being a different protein, or each of these being a different amino acid, if I put them in a different order, that's a whole different protein. Because nature only has 20 ingredients to work with, and so it has to, remember, structure determines function. The way that things are put together determines the job of that chemical. So again, it's just like if I handed everybody a Ziploc baggie with 20 Legos in it, and I asked you to build something. Well, you're probably going to build something different than your neighbor. You have the same 20 ingredients, but you make something different from it. And so that's how nature can make all these thousands and thousands and thousands of proteins from just 20 different ingredients. Another way that nature changes these proteins up is by changing their shapes. Okay? And again, remember, structure determines function. So when you have different things with different shapes work differently. Okay? So in the case of proteins, there are 4 different what we call levels that you can be. All proteins start off looking like a pearl necklace, and that is what we call the primary protein structure. It's the most basic protein you can be. And again, all proteins start off like this, and some proteins stay like that, and they're going to have a certain job. But remember, nature only has so many ingredients. Nature can only make so many things. So what nature can do is make one protein and then change its shape. And by changing its shape, it gives it a different job. So all proteins are made in this primary protein structure, which is made to look like this pearl necklace. Now, if they just begin to change shape, that's what we call the secondary level. And these are just 2 examples. Some proteins will start to curl up. They'll start to twist. That's what we call a helix. Some of them will start to fold, like we've taken a piece of paper and we're folding a fan. That's called a pleat. Some proteins, maybe the top folds and the bottom stays the same. Maybe some proteins, the top stays the same and the bottom starts to fold. It's thousands of different possibilities as to how these proteins change shape. Now, something's going to have to hold them together. Like if you make a fan, if you fold up a piece of paper, you know it's going to unfold. So in this process, nature makes bonds in between them in order to hold them together. So you're more complex. So a protein that has secondary shape is going to have a more important job than a primary protein structure. Like I said, primary protein structures are your basics. All proteins are important or else nature wouldn't make them. But if nature takes the time to change the shape and make sure the shape stays like that, it's going to be a little bit more important job. Now, some proteins will stop at the secondary level, but some proteins will twist and fold until they cannot twist and fold anymore. They are dense. They are compacted. When they get to that point, that's what we call the third level, or the tertiary protein. And again, not all proteins do this. Some proteins stay primary, some proteins will stay secondary, but some proteins will keep twisting and keep folding until they cannot twist or fold anymore. They are as dense as they can be. Well, if that's the case, you're a tertiary protein. You're going to have more bonds holding you together, so your job is probably a little bit more important than the secondary protein. And some proteins don't even want to stop there. They want to become partners with another protein. And that's what you're seeing in this picture. This is a picture of hemoglobin. And there's actually 4 proteins in this picture. There's a green protein, an orange protein, a red protein, and a blue protein. Well, this is like a supergroup. This is like a partnership. So when a bunch of proteins come together and work together, it's called a quaternary structure. It doesn't mean you have to have 4 proteins. Like this picture here only has 2 proteins, but they're still working together. They're twisted up with each other. They're working together. They're acting as one big protein. So depending on how folded you are, determines your complexity. And so some of the really most important proteins in nature are going to be this quaternary structure that we have here. So we only have 20 ingredients in nature, but we can make millions of different types of proteins simply by changing their shape. And this is just another picture. You can see the primary, the secondary, the tertiary, and the quaternary. Now, in the case of anatomy or biology, proteins that we're going to be looking at, we have structural proteins. These are proteins that are going to somehow provide structure. So a common example is collagen. Collagen is one of the most strongest proteins in nature. It's incredibly complex. It's a quaternary protein. It's found in tendons. Tendons hold muscle to bone. It's found in ligaments. Ligaments hold our skeleton together. It's found in cartilage. Incredibly strong protein. It's also found in keratin. Keratin is what our hair, our skin, and our nails are made of. And just a little side note, keratin is white. Look at your fingernails. Your fingernails are white. Well, your hair is the same color. Your hair doesn't turn white. Your hair is white. It's just nature puts other proteins in there to give you your hair color. And as far as humans are concerned, we don't really need hair color anymore. Hair color in animals is used for camouflage and protection against the sun, which is why we've lost most of our hair because we don't need that anymore. So hair color for us really isn't that important. And it actually gets to a point when your body's like, why am I cooking all these recipes if my body doesn't need them anymore? So your body stops making the pigments that give you your hair color, and your hair is naturally white. So that's when the white starts to show up. We also have what we call functional proteins like hemoglobin. Hemoglobin is found in red blood cells. It helps carry oxygen. You've all heard of antibodies before. Antibodies are proteins that we use as weapons to fight off bad guys. And most importantly, we have an incredibly important type of protein in our body called enzymes. Now, in the case of protein function, again, we'll find them, it helps build things. We find it in muscle, hair, fingernails, and then here are enzymes. Enzymes are what we call a catalyst. And a catalyst is something that speeds up a reaction, but it doesn't change a reaction. Think about a piece of food. You leave a piece of food out on the table. Think about how long it takes that piece of food to rot and fall apart. It takes a long time. It would take a long time for food to break down in our digestive system if we didn't have enzymes. But enzymes are there to help our body speed things up. And that's what a catalyst is. A catalyst is, it changes the speed of the reaction, but it doesn't change the reaction. And you're going to see that enzymes end in ASE. Now remember, if you see OSE, it's a sugar. If you see ASE, it's an enzyme. And the name of the enzyme always tells you its partner. Enzymes are very specific. I always think of enzymes as Pac-Man. So I have an enzyme that's shaped like this. And just like you need to hold something to tear it, enzymes need to hold on to something to help them do their job. And so where enzymes hold on is this little space right here. That's called the active site. So notice this active site is like a triangle shaped. So if there was a particle that was round, this enzyme can't hold on to it. This enzyme can't help it. Enzymes are specific. They only work with certain things. And a lot of times the name of the enzyme will tell you what they work with. So for example, protease is an enzyme that works with proteins. Lipase is an enzyme that works with lipids. And you're going to see lots of enzymes in this course. And we'll go over what they mean and tricks of the trade as we go. Again, I talked about how enzymes are used in antibodies and then hemoglobin and things like that. But like I mentioned, all enzymes are catalysts. They speed up reactions, but they don't change the reaction. And an example I always use is if you were to walk into my classroom and I blindfold you, and then I have a puzzle box and I ask you just to grab one piece of the puzzle. You're blindfolded and you can't talk because chemicals can't talk. And I ask you to walk around the room and put the puzzle together. You're literally bumping into people and you're fumbling around to try to see if your pieces of the puzzle fit together. It would take you forever to put this puzzle together. But if I was an enzyme, I have two hands, two active sites. I could grab hold of one person. I see, oh, these two pieces match. So I can go grab hold of this person and I grab hold of this person. I don't do anything else, but these people are like, oh, when enzymes got me, that means my partner's nearby. And those two people can put their piece of puzzle together really fast. So enzymes are going to help speed things up. And so like what I was saying, all enzymes are specific. And enzymes are divas. They are the divas of proteins. If they're not happy, they stop working. And when proteins stop working, that's what we call denature. When a protein, and my handwriting is not good with this, when a protein denatures, that means it falls apart. It changes shape, it doesn't work right. So if my enzyme has an active site that's supposed to be a triangle, if it gets too hot, it could cause it to shrivel up, and it can cause the active site to close. If it fills up with water, it could cause the active site to get really big, and it's not going to work the way it's supposed to. So enzymes are very, very, very picky as to what conditions they work in. So this is why our body needs to stay 98.6 degrees. If we get too hot, it can cause our enzymes to change shape, and they're not going to work. If it gets too cold, it can cause our enzymes to change shape, and they're not going to work. That's why we have to have the right amount of salt in our body. Okay? Enzymes are the divas. If they're not happy, they quit working. That's how your laundry detergent works. There are enzymes in your laundry detergent, and some of those enzymes work in cold water. That's why you wash your dark colors in cold water, because those enzymes work best in cold water. And this is why you wash your whites in hot water. The laundry detergent has different enzymes for hot water. The same thing is true. Okay? This is why our body has to be a certain temperature in order for our enzymes to work the way they're supposed to. That's why when people get fevers, they begin to hallucinate, or when people, their body temperatures drop, they begin to hallucinate, because the brain proteins aren't at the right temperature and they're starting to denature. They're starting to break down. And the person begins to hallucinate because the brain's not working correctly. And also the brain's trying to say, hey, I'm making you see weird things. Isn't this weird? But the person doesn't realize that that's weird. But in the case of these terms I've been talking about, again, think of enzymes as having specific shapes. So some enzymes might work with round particles. Some enzymes might work with particles that are square shaped. And again, this is, just think of them as shapes. But enzymes are specific. And that little area where you can lock into is this thing called the active site. So you can see right here, the active site. And if you're not the right shape, that enzyme cannot help you. And whatever the enzyme is working with are these things called substrates. Now, some enzymes will put things together, and some enzymes will take things apart. This is just an example of an enzyme that's going to break something down. It's going to take it apart. And whatever it is that it makes is called a product. So we're going to be talking about enzymes a lot throughout the year, and depending on what body system or what process that we're talking about. And again, enzymes are specific. They can only act on one thing. So think of them as having certain shapes. So if the shapes don't match, it's like trying to put pieces of the puzzle together. If the shapes don't match, it doesn't work. Enzymes are reusable. Enzymes are real important, and it's like having a measuring cup. You don't want to keep having to buy the measuring cup over and over and over again. So your body does allow you to use an enzyme over and over and over again, but eventually the enzymes will get old and your bodies will just make new ones. But they are reusable. They don't get thrown away after they get used. And again, I mentioned enzymes are divas. Okay? They have to have certain temperatures. If they get too hot or too cold, they're not going to work. They have to have certain pHs. They have to have certain amounts of salt. Okay? And that's what this little page here talks about. But keep in mind it can go both ways. So this says a decrease in temperature will decrease reaction rate. You can also flip it. Basically, if something is too cold, it's going to slow down the enzymes. Excuse me, I'm hot. If it gets hot, it can actually speed up the enzymes to a certain point, and then the enzymes will start to fall apart. An enzyme will lose its shape, which is what we call denature, in very high heat. It will also lose its shape in really low heat. That's why hypothermia, people freeze to death. Their proteins stop working. If their proteins don't work, the cell doesn't work. If the cell doesn't work, you die. Proteins are incredibly, incredibly important. Extreme pH can slow the reaction. An increased substrate or enzyme concentration can increase the reaction. You can flip that around. Decreased substrate or enzyme concentration can also decrease the reaction. But we're going to be doing a lot with enzymes throughout the year. And so again, when we talk about enzymes denaturing, you can see they change their shape. And remember, structure determines function. So the shape of a protein gives it its job. So it's like me having a car. If I start to take the car apart, it's not going to work the same way. Okay? So if I start to change the shape, it changes the job. So that protein's not going to do the job it's supposed to. The cell's not going to do what it needs to do, and that cell's going to be damaged and probably die. Just another picture. You can take a moment to pause to see how enzymes work. Which leads us to our last thing. And I'm just going to be brief on this one because we're going to have PowerPoint presentation videos of just nucleic acids. But nucleic acids are the fourth on our list because they have the most parts. Remember, carbohydrates are carbon, oxygen, hydrogen. Lipids are carbon, hydrogen, oxygen. Proteins are carbon, hydrogen, oxygen, nitrogen. This guy has a fifth one. CHOMP is what we say. Carbon, hydrogen, oxygen, nitrogen, and phosphorus. Now I have even more Legos so I can build something really big and really important. And nucleic acids are DNA and RNA. DNA is, as you know, your genetic material. DNA is your cookbook. DNA holds your recipes, which are your genes, and those genes tell you how to make proteins. And the Legos that we use to make nucleic acids are called nucleotides. Now remember, for carbs, they're monosaccharides. For lipids, it's fatty acids and glycerols. And proteins are amino acids. In the case of nucleic acids, it's nucleotides. And you're going to see, remember the number 3. The number 3 is incredibly important when it comes to nucleic acids. DNA has 3 letters. RNA has 3 letters. There's 3 parts to a nucleotide. There's going to be 3 parts in making a DNA. There's 3 differences between DNA and RNA. There's 3 kinds of RNA. There's 3 steps of making RNA. There's 3 steps of making a protein. But when we talk about nucleic acids, we talk about these things called nucleotides. And I always draw a nucleotide as an L. It has 3 parts to it. It has a phosphate, it has a sugar, and it has something called a nitrogen base. So this is my Lego. And so I'm going to stack these Legos up. So you can see right here my 3 parts. The circle is a phosphate. The pentagon is a sugar. And these things with letters here in the middle are my nitrogen bases. And if you think of DNA as a cookbook, you're looking at a book here. The sugars and phosphates make up the book cover. And you know the book cover's job is to protect the recipes on the inside. The nitrogen bases here are your genetic material. So when we talk about genes, we're actually going to be talking about the nitrogen bases. But again, we're going to do a lot more of that later on. So when we look at nucleic acids, again, this is your DNA and your RNA. They store information in the form of a code. And we're going to talk about that code, and you're going to learn how to read that code. And DNA stands for deoxyribonucleic acid, and it contains the instructions to make all the proteins that your body needs. This is your cookbook. RNA is ribonucleic acid, and its job is to help make the proteins. And again, we're going to talk about the 3 parts to them. We'll talk about the 3 differences. We'll talk about why we need RNA, where RNA is allowed to go, where DNA is allowed to go, and all that stuff.